Let's talk about the smallest components of a muscle fiber, organelles. Organelles are going to be a little special, a little different in a muscle fiber than they are in a generic cell. Muscle fibers are a lot longer. They kind of have a spaghetti sort of shape to it. They're going to stretch from point A to point B and usually connect to another muscle fiber. And we're going to have to keep them separated, and they're going to be in their own little sort of fluid themselves. We're going to dive into these two specific organelles a little more in a second, but let me just kind of give you a locational area for them. So there's the sarcolemma, which is the cell membrane of a muscle fiber. And you can see that it sort of goes all around the cells, and it sort of holds these muscle fibers or filaments together. And then the liquid that surrounds these guys are going to be the sarcoplasm. So they sort of remind you of cytoplasm, maybe. Sarco is the muscle, so sarcoplasm. It's going to surround the cells. And they prevent friction, but they also have a lot of good stuff that the muscle needs to keep it going. So let's talk about the sarcoplasm, that liquidy layer, first, and then we'll talk about even a new organelle that we're going to cover. The sarcoplasm holds the cells' molecular movements together. It kind of is specialized because it sort of is that liquidy layer that's there. It holds a lot of glycogen, which glycogen is just something that stores a lot of energy. And it also has a lot of myoglobin, and myoglobin is something that stores a lot of oxygen. The two things that muscles need the most of is going to be energy, oxygen, and ATP. And we'll talk about those a little more soon. The other specialized organelles that we're going to find in this muscle fiber are going to be these things called myofibers. They're long protein cords that run through the muscle fiber, and we're going to see a little more about them soon. And then they have their own different type of ER. It's going to be called the sarcoplasmic ER, and it's a smooth endoplasmic ER of the muscle cell. And what this is going to do is it's going to have reservoirs of calcium, and it's going to basically let calcium flow every once in a while to let the muscle actually move itself. And the sarcoplasmic reticulum is going to crisscross around the muscle itself, and it's going to allow it to either release calcium and sort of let the muscle move, which we're going to learn a little more about in a second. And then finally, we have these transverse tubules. Remember, transverse just means across. And so these are going to be these tubules that extend from the sarcolemma and penetrates each of the cell. And why we need to have these special tubules is because when there's an electrical current coming from the outside of the muscles, it can kind of penetrate into all of the cells and tell the cells to work and coordinate their reactions. If it didn't have this sort of thing, each muscle would have to have their own stimuli saying, do this work, don't do this work, and that would be a lot. Alright, let's go ahead and look at a quick anatomy. We're going to label and talk about all these different things in an upcoming video, but what I wanted to kind of just give you an idea of is just some of the parts. So we know that we have a larger muscle structure, and we've now sort of looked at just one of these tubules, and that's going to be our myofibril. And if we look at the myofibril up close, what you're going to see is, number one, this blue thing in this picture, that is going to be that ER, the sarcoplasmic ER that we talked about. These little things over here, you see a lot of them. Those are going to be my mitochondria, and they're going to produce a lot of energy and ATP for us. You'll also see this sort of band that's going around. It's transverse. So that's going to be our transverse tubule. You'll see that it has little holes in it. See, there's a hole, there's a hole, there's a hole, there's a hole. And what they're going to do is they're going to make it so if this muscle hits with some electrical current, all the little cells will get all that electrical current, so they get the stimuli at the same time to do whatever they need to do. And they activate themselves not just by electrical current, but by calcium. And these guys that sarcolomic rough ER is going to, sarcolomic endoplasmic reticulum, excuse me, it's going to release calcium into these channels and allow the muscle to sort of coordinate and move. The muscle also has a really special type of proteins, and these proteins allow the muscle to sort of expand and stretch out, and then also condense and become really small. So they're going to need this ability to sort of crawl and become small and crunch up, and then also sort of expand out. When you pull your arms out as far as you can, you're stretching your muscles as much as you can. But then, of course, when you sort of scrunch up to become a little ball, you're not losing cells. They have to sort of condense, and this is going to be the process of that. There's going to be these different things that are going to help move, open, and close them, and we'll see that picture in the next video probably. Myofibrils are long protein cords that run the longitudinal process of the muscle fiber, and they contain these things called myofilaments. Notice the pattern, everything has myo in it, muscle, and these guys are made of proteins. There's a thick myosin filament, and there's a thin myosin filament, which we call an actin filament. So now we're really looking, just sort of to give you a roadmap of where we are, we're still looking at one of these really tiny, tiny little cells, and we're looking how each one of those cells work. And again, we talked about the thick and the thin, but let's just kind of give it a little structure and a little picture of what they are. The thin microfilaments are made up of that myosin. Each contain these intertwining little heads. Can you see these little two-headed things that sort of pop up? You have this filament, this tail, and then this ATB actin binding site. And basically what they're going to do is crawl on this thin head. And the thin head is made up of actin, that's the little blue balls, and they contain these active sites where the myosin head can sort of attach themselves. So see how they have this sort of like cup in them, and this sort of has that sort of adapter, and they sort of fit in together. And it allows these guys to sort of crawl, to either condense the muscle to be shorter or to make it as long as possible. So just remember, thick portion, thin portion. Actin is going to be the thing that's over here, and this is going to be myosin. And ATP bindings is going to be the thing that is going to make this guy move, but we'll talk way more about that soon. So this is how they work. This is kind of what they look like in every cell. You'll notice that, number one, you have that thick myofiber. And remember how they're made up of that myosin? They've got those little heads on them, those globular heads, and they sort of fit into our thin myosin fiber, and that's the actin, and they'll be able to sort of crawl this way, and when they extend, they actually go this way as well. So these little things are going to actually move across and crawl over this thin myofilament layer, and they do this with these little heads. The heads will sort of crawl, 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 and when they do, these two areas are going to condense, and then eventually, if we need to stretch, they're going to go ahead and crawl, crawl, crawl in the opposite direction, and then you'll see this area of the muscle and this area of the muscle go in the opposite direction. They'll pull apart from each other. And that's called the sliding filament. We're going to talk way more about that soon. There are some regulatory proteins, and these proteins are going to control how the muscle works. And really what we're going to be controlling is this thin myofiber. And you'll see that there's a couple things going on. There's a tryptomyosin. So the tryptomyosin is this cord that's going around, and see how like those little holes, remember that's where the actin, those globular components of the thick myofilaments come together? Well, they're blocked right now, which means that if this is right here, that tryptomyosin, there's no way that muscle could crawl across this structure. So if we want to make it so this muscle doesn't move, we're going to leave this filament in place. But if we need to get this muscle to move, we have to actually move this cord out of the way. It's almost like a safety feature. We've got to move this out of the way to expose all these little holes, so that way the globular heads of the thick microfilaments can actually crawl in them and either condense the muscle or contract the muscle. So remember the cordy thing is called the tryptomyosin. It wraps around the actin and controls whether the myosin head can bind. The way that we can get them to unbind is when calcium binds to these yellow things called triponin. The triponin will actually go ahead and shift over. And when it shifts over, it also moves the tryptomyosin away, and then these little holes are exposed. And when that happens, it causes the ability for those myosin heads that are on the thick microfilament to be able to attach and move.